Okay, so for today's demonstration, we are going to be going through setting up an enzyme assay. The enzyme assay that you guys are going to be asked to do in your exam next week is going to be assaying the activity of cellobiase, similar to what you've been doing in lab for the last few weeks. Now, as you know, if you need to determine the purity, the fold purification of a purified sample of enzyme versus the crude, there are two parts to the experiment that you need to do. First off, you need to assay the enzyme activity, and you also need to assay the protein, the total protein content. We're not going to go through that in this particular demonstration. Assaying for total protein is the same as the last video that we did, the Bradford assay. It's going to be the exact same. You're going to go through it the exact same way. Now, the important thing to remember is that whatever you do for the enzyme assay has no bearing on what you're doing for the protein assay. Make sure that you consider them completely separately. So any dilutions that you do for the enzyme assay, there is no reason to expect that any sort of similar dilutions would be appropriate for your Bradford assay. So just approach each one completely separately. And then you can compile your data afterwards in your purification table, work out your calculations, and determine your fold purification, which is your goal when you're doing this particular task. Okay guys, so what we're going to be starting with at the start of our experiment are samples of two other solutions. So I have a crude sample and a purified sample here. One of the important things to remember here is that these are just samples. These are not the total fraction volumes as we've been referring to them in our experiments in lab over the past few weeks. These are just fractions of them. So you will need to look at the directions that you've been given. If you don't have the total fraction in front of you, have a look and see if you can find information in the information you've been given on what the total fraction volume was. Like I said, this is only a sample of that. Now we can still determine a lot from just these two samples. So I have my crude and my purified. Again, I'm just going to be doing the enzyme assay right now, and the way that the enzyme assay is set up, essentially what I need to do is measure the appearance of product. When you're looking for enzyme activity, there's essentially two things you can do. You can do, number one, look at the disappearance of substrate, and number two, you could look at the appearance of product. That is the only way that we can measure how much work the enzyme is doing, because we can't see the enzyme working itself. So we're going to measure the appearance of product. I'm not going to go through the biochemistry behind how cellobiase works, so make sure that you review that. You should be familiar with that. But because we've been doing this assay for a number of weeks, you'll be familiar with the substrate, which is p-nitrophenyl glucopyranoside. What happens is that cellobiase will cleave this product. We'll have a glucose molecule and a p-nitrophenyl, and p-nitrophenyl is actually what we're going to measure. So, depending on the amount of enzyme that's present in each of these fractions, if I give them the same amount of substrate, I can then measure the amount of product that's made and determine how many units of activity that I have. So, the first thing that I'm going to do is think, well, I have no idea what concentration of enzyme I have here, if it's super concentrated. What I do know is that my enzyme needs to incubate with the substrate for 5 minutes. Now, in the interests of saving time, I'm going to think, well, there's no reason for me not to set up an assay with the fraction as it is right now, and also with a 1 in 10 dilution of the fraction. There is no reason to not do that. And so I'm going to go ahead and make 1 in 10 dilutions of each of these two fractions here, my purified and my crude. So I'm going to get a couple of microfuge tubes here. And as always, I'm going to label them before I put anything in there. I'm going to call this one, and just label them pretty much the same way that they're labeled in the original in the original tube, and then just add a 1 in 10 so that you don't get confused with what's your diluted sample and what's not. 1 in 10. Okay. Now, when I'm setting up my assay is going to be a 1 ml assay. I'm going to have 900 microliters of substrate, and I'm going to have 100 microliters of fraction, which contains my enzyme. So I need to be able to add 100 microliters to this assay. So when I'm thinking about making up my 1 in 10 dilution here, I don't just want to make up 100 microliters of this, because if I have 100 microliters in this tube, if I put 100 microliters in, I won't be able to get 100 microliters back out of it. It's just the, I like to call it giving back to the universe. The universe will take its cut. You'll be a couple of microliters short if you do it that way. And so instead I'm going to make up 200 microliters of each of these. And so I'm going to start by putting in the small volume, and then when I put in the diluent, that larger volume will actually mix through the small volume for me. So let's go ahead and start this. So I'm going to be pipetting 20 microliters, so I will use my P20. And again, review the other video, guys, the Bradford video, for tips on proper pipetting technique and organizing your workspace and all that fun stuff. Okay, it's probably, so here we go. This is my P20, it's wound to 20 microliters. Going to, and again, remember, as I said in the last video, when you're going to be pipetting, lift up the tubes. Don't be doing this too low, too far away from you, because you won't be able to see accurately what you're pipetting. Try and make the OK sign with your hand here so that you can see through the tube. You're going to pipette 20 microliters, making sure not to totally dunk that tip in there. I'm going to add this, and I'm going to add this directly to the bottom of the tube so that I can see exactly where the 20 microliters is going. Try not to just pipette it loosely into air. And the reason for that is that this is a really small volume, and so if I lose 2 microliters of the 20, that's 10% error right there, and that's an error that I can't really afford, so I just want to pay attention to that. I'll take 20 microliters of this one. And in here, there we go. Now I need to add 180 microliters of diluent, so for that I'm going to use my P200. And the diluent that you use will depend on what your fractions are made up in. You should use a buffer that's going to be most similar to what your fractions have been made up in. So depending on if it is an ammonium sulfate precipitation that was carried out, or if it was a homogenization buffer or a gel filtration buffer, that's the buffer that you should use for your dilutions. Now in your exam, you will be provided with the appropriate buffers, but make sure that you spend a little bit of time thinking about why you're using particular buffers. And so here is my buffer. Okay, so I've got 180 microliters, and again, I'm being careful here not to totally dunk my pipette tip. Obviously it needs to be below the surface, but try and avoid dipping it too low into the solution, because you'll just get extra drops around the pipette. Okay, I'm going to put 180. And if you like, you can gently pipette this up and down a couple of times. You're pipetting quite a large volume in, so it won't take too much to mix it. But just be careful when you're doing that, and always remember that you're trying to avoid generating any bubbles in your samples. You of course need to change pipette tips even though you're pipetting the same solution, because what's in my microfuge tubes here is different, and you want to always be conscious of where cross-contamination can occur. Okay, so I have both of my dilutions made up. Now I need to set up my enzyme assays. So I essentially have four samples that I'm going to be assaying. I have my crude here, the original, the undiluted, and a 1 in 10 dilution, and then my purified and a 1 in 10 dilution of that. So I'm going to need 4 assay tubes. So 1, 2, 3, 4 for each of these. And then one more, which I'm sure all of you were thinking, what about the blank? So this is for my blank. It's helpful to think of the blank for this particular experiment, for this assay, as being a negative control. it is really important that you do everything to your blank that you've done to your samples, because otherwise if you see enzyme formed in one of these tubes, which are then going to go sit in a 50-degree water bath for 5 minutes, and you haven't subjected your blank to that same 5 minutes at 50 degrees, well then I'm just going to say, well, the reason that you saw colour development here, the reason that you saw product appear was because you had it at 50 degrees versus you didn't have it for your blank. So just, if it helps, think of your blank as a negative control. You must do everything to it that you do to your fractions. It just should not have that one thing that you're testing. So we just don't want to put enzyme in here, but everything else will be the same. So what we're going to do is get some substrate here. Where is the substrate? Here we go. And I'm going to put 900 microliters of substrate into each of these tubes. Now, I just mentioned a couple of minutes ago that I could put the small volume in first, followed by the larger volume. So that would be implying that I'd put the 100 microliters of this in first and the 900 in on top of it. The reason that I'm doing the 900 first is because this is actually going to save me time, because I don't now have to change pipette tips at each point. Because there is nothing in this tube, it is a clean tube, there is no risk of cross-contaminating anything at this point. And you'll notice I'm being quite careful to make sure that I'm not losing a drop along the way. Okay, here we go. Now before I put my fractions in here, I'm going to make sure to label things up. It's really easy to get distracted for a second and to just forget what went where. Let's put the lid on this substrate so we don't spill it. Okay. Just gonna label these with the purified fraction, and then 10 diluted, crude, and the crude, 1 in 10. Okay, and now I'm ready to add 100 microliters to each of these, and so I will need my P200, of course. Wind it to 100 microliters. There we go. And I'm just going to move. I'm going to move left to right on this through the samples. And if you remember from when we did the Bradford video, all of my fractions here are in the front row of the rack. I'm going to move them back one as I take the sample from them. And that will mean that if something comes up, if something distracts me, I'll know which fraction I stopped on, and I won't have to think, did I add this fraction to the assay already? So, put that in there. Also moving my reactions back one row. And if you get in the habit of doing this, it just means that if you get, like I said, if you get distracted, if somebody starts talking to you or asks you a question and you turn back to your experiment, you'll still be confident that you know where you're picking up. Okay. So, I've added my fraction to each of these samples here. They're all labeled. I'm going to give them a quick inversion here, each of them. Typically, you want to be pretty quick about the start of your reaction. So we've got a clock going now from when the reaction started. So I'm going to go ahead and put these into the 50-degree water bath for 5 minutes. Let's just go over here. Okay, these samples need to incubate, like I said, for 5 degrees, or for 5 minutes at 50 degrees. You have a little bit more leeway here in terms of the start time of the reactions. For example, this reaction where I added this, my first sample, this would have started maybe 30 seconds ahead of this reaction. typically, if you're doing a 1 minute assay, you'd be quite concerned with that difference in time. But given that we're doing this for multiple minutes, for 5 minutes, and some of you may have done even longer assays over the last couple of weeks, the longer the time, the shorter or the smaller the error is in terms of however long it took you to set those up. So you just need to sort of think about it on a case-by-case basis as how important is it that I'm really, really fast in setting up this particular assay or this particular test. So you do need to think about it, because it's not going to be the same for every single assay that you do. When we were doing the LDH assays, time was much more important because the assay reaches equilibrium incredibly quickly, and so we need to be able to determine the initial part. And so you'll remember when we did the LDH assays, we didn't add the substrates and the enzyme together until right when we were ready to measure the absorbance. So we don't need to worry about that quite so much at this point. The other thing to remember is the time. The important thing here is that all of our samples incubate for the same amount of time. If you let your samples incubate for a minute longer, that might not be a disaster for your experiment, as long as you know the amount of time that your assay was incubating for. If it's 6 minutes, that's fine, as long as you know it was 6 minutes and you don't then take 5 minutes forward into your calculations. Okay, so as this is incubating, I'm going to set up my cuvettes to be ready for when my assay is done. I want to be able to stop my assays after 5 minutes. As you guys know, the product p-nitrophenol that we're going to be measuring is not visible. It doesn't have absorbance within the visible spectrum at the pH at which the assay is conducted, which is pH 5. So we need to raise the pH, so we use stop solution for that. The stop solution does two things. First off, it stops the assay, which means that I've got a little bit of breathing time while I set up to read my assays on the spectrophotometer. And the other thing is this stop solution is at pH 9.5. And so it will allow me to visualize the p-nitrophenol. So I'm going to put 1 ml of stop solution into the cuvettes that I have. And I'm going to need, you know what, before I do that, of course, I should probably label these up. I'm going to need 5 cuvettes, so let's just put these to the side. I'm going to go ahead and label these. And again, as always, try and minimize your handling of cuvettes, guys. Make sure that when you handle them, that it's always going to be near the top. Any sort of writing on them should go near the top. And use the same labeling that you used on your assays. So purified 1 in 10, crude 1 in 10, and blank. Okay, so I'm going to put 1 ml of stop solution into each of these. Let's just verify that's 1 ml. And be careful again when you're pipetting 1 ml, if you aspirate too quickly, it's easy to get bubbles. Again, I can keep the same pipette tip here because the container that I'm pipetting into is clean. And you probably can't see at this distance, but I'm touching the pipette tip to the edge of the cuvette, the inside of the cuvette. And what that does is it draws out the last of any liquid that's in there, and that will just help you be more accurate in your pipetting. Okay. Setting these up ahead of time, and if you're concerned, you can set these up even before you set your assays up, but setting these up ahead of time means that once my 5 minutes is up with my samples, I can just bring those back over here, and I'm going to pipette the entire reaction out of those tubes and into these, and it will be stopped immediately. The product then, p-nitrophenol, is going to be stable for some time. You don't need to rush to get the samples specced. The important thing is when you stop them, and that you're stopping them, you don't just stop 2 and then, you know, go answer the phone because it's been ringing and then come back and stop another 3, because obviously the time difference between those is going to cause variation in your results. Okay. Okay guys, so the 5 minutes has elapsed, and I've just removed my samples from the water bath. I actually took them out at around 4:45. I'm going to take them out of this floating water bath, put them back into my rack in the same order that they were in. And the reason for this is that I want to pipette them out of the reaction tube and into the cuvette in the same order that I set up the reactions, because the delay between one and the others will be sort of mimicked in the delay here, and it should just minimize error. So I know my reactions are 1 mil reactions, so I'm going to get my P1000 still wound to 1000 microliters. I'm going to pipette the entire assay out, and you'll want to be careful about doing this, guys. There's only 1 mil in this tube, and so if you're not careful, you may miss a significant portion of it. Okay, so the first thing we noticed here, guys, is going to be the difference in color. As soon as I pipetted these into the cuvettes, I can see that there is variations in different colors of yellow, and that's what I would expect to see when the product is formed. The next thing I'm going to notice is that the colors of the undiluted samples are very, very intense yellow, and they are going to be too yellow to be able to read with any degree of accuracy on the spectrophotometer. So at this point, I'm pretty glad that I did set up my dilutions as well at that point. Now, I'm not going to go through how to set up your spectrophotometer and all that fun stuff. It is the exact same as the way that we talked about it for the Bradford assay. You're going to calibrate with your blank, and you're going to configure with one of your samples here. This is not going to take near as much time, however, because of course we don't have a standard curve here. We're going to determine concentration using Beer's law. So again, make sure that you review your directions for instructions on how to do that. That's going to be pretty much it. This portion, setting up this part of the experiment shouldn't really take you very long. As you can see, pretty much you're just going to set up some dilutions, you're going to set up your incubations, and as long as you are organized and you plan ahead of time what you're going to do, then there shouldn't really be any issues, and it shouldn't really take you any more than, I would estimate, around 10 or 15 minutes to do this. That's going to be a good guideline for you when you're doing this in your exam. Think about how long should it take me to do this, and just, use it as a guideline. If you go a little bit over, then that's fine, but you do want to be a little bit cognizant of the time, because that is one of the things that you are being assessed on.